Navaneel Sarangi. I hope I'm pronouncing it well. I'm sorry if I'm not. I'm doing my best. He's a graduate student, and also I believe he has been a research associate at the Marvel Space Institute of Science, and he will talk about radiotolerant mammalian cells under low Earth gravity, Earth orbit conditions.
So is my screen visible?
Yes, it is.
Okay,
Go
thank
ahead.
you. So yeah, so the first speaker kind of paved the ground by mentioning the word radio protection and how galactic cosmic radiation is one of the biggest problems that we're fighting in space exploration. So today my topic is radio protection in low Earth orbit for human cells using tardigrade proteins. This is a particular project which I'm working on with Dr. Siddharth Pandya of Blue Marble Space. So let's start. So just a second. Okay. Uh, so today we will discuss why this topic is of importance and what literature basically says about this topic and what we propose to do about it and what is the novelty behind this particular idea. So in the first place, why do we need a radio protectant? Space as it is a very hostile environment and it is full of stressors of which radiation is possibly the most important factor. This radiation may be coming from the galactic cosmic rays, radiation, the solar particle events, or radiation from the Van Allen belt constraints as well. And these have chronic or acute effects in response to its interaction with the biological samples. This leads to various radiation-induced disease in the space travelers and has the capability to affect the immune system, the neural system, the cardiovascular system, and can result in cancer as well as advanced aging. It is a very persistent threat to continued exploration of outer space as well as its habitation. But why are we in the absolute dire need of a radio protection? Why is it now that we need it? Well, of all the radiation sources responsible for the genome mutagenic changes, galactic cosmic ray is of the highest concern, and it is essentially composed of several high energy particles originating from various astronomical events. Among these, there is the presence of high charge and energy nuclei, which are particles having high linear energy transfer and can have significant impact on biological tissues when they interact with them. On the other hand, the solar energetic particles, or SPE, have the origins in solar flames or coronal mass ejections and comprise low to medium energy protons. Why is GCR so dangerous? Well, in the first case, they consist of 8Ze particles, which are a form of ionizing radiation. These ionizing radiations can have direct impact when they interact with the DNA and can cause DNA damage or indirectly through the production of free radicals like hydroxyl. And when looking at the present modes of protection, which is passive and active methods, whereby the word passive basically means the lead protection and active resistance means the electromagnetic modes of resistance which are being looked into. None of these actually comprise a holistic protection against all of these radiation sources. So even with all of this radiation, NASA estimates that for a journey to Mars and back, a particular astronaut will receive around 660 millisieverts of radiation when the career limit is about 800 to 1200 millisieverts in the first place. Therefore, when Hashimoto and Kuniada et al. Discovered the protein DSUP and the fact that it could actually protect mammalian cells from X-ray damage, it was an amazing discovery. Once this discovery was made, this particular protein was taken and expressed in plant systems as well, MD simulation done of it, and the results are wonderful, which I'm going to discuss right now. At the end, if we can prove that this radioprotectant can be expressed in a human being and it can produce radioprotection, then it can be used in the field of medicine, nuclear energy, and of course, in space. So how will we do this? In current literature, there are several methods have been proposed already, and the first speaker talked about the hibernation aspects of it. And in this case, in my case particularly, we are going to talk about radioprotectants. And of these, our approach using DSUP covers the radioprotectants which are executed using exogenous transgene expression. So the origin of DSUP is from an extraordinary animal called tardigrades. The tardigrades are famous for their resistance to the extremes of conditions, and it is a true polyextremophile. For us, the most intriguing characteristics is actually its resistance to radiation. They can not only survive in simulated radiations worth 5,000 gray, but can actually continue proliferating after exposure to GCR. So taking all of these factors into consideration, we are now going to discuss some literature regarding it and how are we going to develop our project and why is it relevant basically. So the first paper that we come across is from Johnson et al, which refers to an experiment which was conducted in 2007, which revealed that tardigrades actually survive exposure to space environment. And the graph which you see over here actually shows the survivability of two different tardigrade species. And not only did these tardigrades survive, they also came back to it and continued to proliferate. The core of my idea actually comes from this particular paper, which was published in 2016 by Hashimoto and Kuniada et al., when they sequenced the genome of Ramazzottius varieornatus, which is a type of tardigrade. And this revealed various clues to the methods of stress relief, which is present inside the tardigrade. But of importance was the damage suppressor protein, or DSUP. It was found to be present in the nucleus and had abundant expression in the embryonic stages as well. This particular study suggested that the signal sequence which causes it to be present in a nucleus was present in C terminal, and this C terminal of the protein was extremely basic in nature, suggesting that it could have possible interactions with the DNA through electrostatic interactions. So the authors also did a particular study where they injected the DSUP into HEK293 cells, the gene basically, and saw its interactions with X-ray, what protections it could produce inside the cell. What we observe is amazing. For in case of DSUP cells, after 1 hour of radiation, in this case, what you see here, gamma H2AX is actually a biomarker for double-stranded breaks. So if there are more double-stranded breaks, there will be more gamma 2AX. In case of this orange bar, which is for the DSUP protected organism, the gamma 2AX was 40% lower, which is a huge amount of protection which this particular protein is giving to the particular cell. Not only that, after significant amount of radiation, which is 4GY, 4 gray basically, which is actually sublethal to human cells, these cells not only survived it, but actually continued to increase in cell number. And this was an amazing discovery by them. So after this, in 2019, Chavez et al. Did a biochemical study of this particular protein and found some very important facts. It was discovered that the DSUP basically binds to the nucleosomes rather than the free DNA. There can be multiple units of DSUP aggregating at one particular nucleosome. The DSUP does not impede with the folding of the chromatin itself. It binds irrespective of that. And when these particular nucleosomes with DSUP bound was exposed to the presence of hydroxyl radicals, this DSUP actually protected the particular structure from degradation. And this was also amazing. The one more important thing which Chavez et al. Discovered was that there was a particular homologous sequence inside this particular protein, which was same as the human high mobility proteins, which is present in the chromatin structure itself. This was an amazing discovery as it is. Following this, Hashimoto and Kuniada et al. Published a review on this particular protein only. And if you see on this side, this is how the DSUP protects the DNA by forming a shield around the DNA and basically giving it protection. And so this particular research was followed on by a computational experiment, basically MD simulation, which gives us some amazing insight in how this particular protein looks like, how this particular protein occurs. In the previous paper, Hashimoto and Kuniada et al. Actually suggested that this particular protein does not have a particular structure. It must have a free structure, which you can see over here without any strict domains or structures like that, so that it can flexibly attach to the DNA wherever and however the DNA is particularly bending and interact with it. The C terminal of the particular protein is the part which interacts with the DNA actually. And this particular study not only published that as a fact, but also established that there must be the existence of particular electric fields between the DSUP molecule and the DNA structure. This is of extreme importance because in case of active protection against radiation, electromagnetic fields can actually provide a very prominent shield against ionizing radiation. So this might be a very contributory cause to why DSUP provides such great protection against ionizing radiation as well. And this particular study, with all its fact, actually proved that what was found before was absolutely correct in silico method. And in this particular paper, which is still in peer review, it is still in a preprint. and has not been published yet, we actually find that this particular protein can be expressed successfully inside human cells as well. And not only that, they have excellent characteristics for protection against different kinds of radiation as well. Now, what do I propose to do with this particular project? This particular project, I've divided it in 7 phases. We are currently in phase one, where we are doing the literature review and building the contacts, as well as constructing the basic necessities that we will require. But following on, we will actually do a standardization of the particular cell line that we'll be working with, the HEK293 cells with the different kinds of gene constructs, basically construct the DSUP, the promoter, the expression profile. Everything needs to be standardized before we move on. So that will be the second phase. The first experiment that I will be conducting is actually studying the proliferative activity as well as the genomutagenic changes which are taking place inside the protected mammalian cell in the presence of only galactic cosmic ray radiation. And the second experiment will be, well, as you know, microgravity can play all sorts of havoc inside a cell, including changes expression profile. So I need to make sure that when my experiment is going on in low Earth orbit, there is expression of DSUP. And in that case, we'll need to standardize this particular cell line in the presence of microgravity. Therefore, the second experiment will be to characterize this particular cell line in the presence of microgravity. Moving on into the third experiment, it will be actually a point where both microgravity and radiation meet, and the particular cell line which I have characterized and standardized in radiation and microgravity separately will be exposed to microgravity and radiation in the same particular experiment. This is actually a pretty difficult experiment to do, to be honest, but let's see what happens with that. Of course, the last phases, the 6 and the 7, are the most exciting phases, where we'll have to design a payload to study mammalian cells in low Earth orbit. What are the parameters which we are going to use, for which Dr. Siddhant Pandya is working actively with me on this particular project. And of course, the last phase is to launch our project into low Earth orbit and see the changes, and if indeed my hypothesis is true that DSUP can provide radio protection to mammalian cells. So what is the novelty in my hypothesis? Why is this of such importance? Well, as you know, there are several radioprotectants available. Why are we choosing DSUP? Well, DSUP is already present in an organism which has survived exposure to the conditions which are present in low Earth orbit. They have already survived the condition of galactic cosmic radiation. They have already survived in space vacuum. So we have a particular protein which has already done that, and we have the proof for that. Not only that, this particular protein has already been expressed and studied to some extent, not completely. There are several things we don't know about DSUP yet, but we know that it is possible to not only express them in mammalian cells, but also to produce a stringent amount of radiation protection, to get us a stringent amount of radiation protection from this particular DSUP molecule onto the mammalian cells as well. And not only in this case, but in entire plant systems as well, this has been worked out that this is exactly what happens as well. And this particular molecule provides protection against both low and high doses of LET radiation, as well as chronic and acute exposure to different forms of radiation as well. Up till now, majorly we have noticed X-ray radiation, but the study which is in preprint right now actually exposed them to some sort of ionized radiation as well. Therefore, we have this perfect protein which has been proven already to be expressed in outer space, and can be expressed in mammalian cells as well, and can produce protection in mammalian cells as well, which makes this protein so important and so interesting to work with. Now, the future goals of this particular project includes developing a consumable form of the radioprotectant so that it can provide prophylactic protection to astronauts as well as plant systems in space. Because even if we can grow plants in space while traveling in deep space, they will also be irradiated by galactic cosmic radiation. So we will have to protect our food systems as well, and this can also provide protection to human systems as well. So developing a consumable radio protector might be a very good idea to go about with. There are several gene therapy aspects as well. As I mentioned, in the plant systems, we can have gene therapy. We can create transgene. We can create genetically modified organisms which can survive in high doses of radiation and still give us the results that we want. And this particular protein can provide holistic protection against space radiation as well. So that is my project, and that is what I envision to do. Thank you for being such a patient audience today, and you can reach out to me on my email address, my Twitter, and of course, open to any collaborations at all. Thank you so much.
Thank you, Navaneel. Thank you for your presentation. It was really interesting. Unfortunately, I can't see any questions on our chat, but I have one of my own because there are some other species that are resistant to radiation, right? Like Deinococcus radiodurans or some species of fungi. What is the advantage of studying tardigrades over other species?
Yes. So if we talk about Deinococcus radiodurans, we also have to keep in mind that we want to express this particular protein inside a mammalian cell. So the closer the organism is to a human being, the better will be our chances of getting a particular protein which doesn't hamper its natural expression, and which can actually get expressed inside a mammalian system. In case of Deinococcus radiodurans, we have a very innovative system by which they protect their DNA. They have a very high functioning way of repair mechanisms as well. But in case of this particular protein which is coming from tardigrades, they don't do any repair. They simply do, they are basically like a shield. They protect against everything. They're basically a holistic protection against everything. And even if the particular human cell doesn't have very good mechanisms of repairing itself, it will still then get protected. And not only that, we also have to consider the different proteins which get involved. This is not yet studied yet, but DSUP can also provide protection to particular proteins, do some epigenetic modifications in those proteins to provide them with this particular characteristics as well, which is not very well studied yet. So these are the very novel aspects of DSUP, which is very interesting from the other organisms which are present right now.
Okay. I have also another question, thing I didn't maybe get from your presentation. Is DSUP gene present in our genome already, or does it require transfection?
Yes, so that is actually a very interesting question. So in the first experiment, which was conducted by Hashimoto and Kuniata et al., they actually did not find any homologous sequence of the DSUP gene in our genome actually. But when Chavez et al. Did the same thing, they actually found a very similar sequence, which I mentioned, the HMGN protein, which is actually present in our chromatin as well, the high mobility group proteins. So they actually found a similar sequence from the DSUP gene in our genome as well. It was a very short sequence, but this sequence is very essential since this is the sequence with which the HMGN protein in our body actually binds to the nucleosomes. So it was very interesting to find that the DSUP, which was a completely different organism, which has nearly no evolutionary similarity to us, also had that particular sequence which was actually conserved, and that was the only similarity between these two genes. It is not present in us.
Okay, thank you.
Thank you.